Most species live, age, and fade on a schedule shaped by food, predators, and genes. But evolution loves variety. In a few lineages, the clock runs dangerously slow, or can be wound back entirely. These are not superheroes. They are animals tuned to cold, stability, protection, or radical cellular plasticity. Their life histories are natural experiments that answer one question better than any lab. What happens when you remove the penalties that usually force death? We examine those experiments. We'll start with a landmark comparative study that finally put cold-blooded animals under a systematic microscope, then dive into the deep with Greenland sharks, drift among ancient black corals, and brush the silica spires of glass sponges, and end with the jelly that, in the lab, chose to become young again. Along the way, we'll ask, is immortality ever truly immortal? And what can these animals teach us about aging, conservation, and fragility? A global study that changed how we think about aging. For decades, biology textbooks treated aging as nearly universal. Little creatures live fast and die young. Big, warm-blooded animals lag behind. But a massive international effort assembling mark recapture records from 107 populations across 77 species of wild reptiles and amphibians blew a new wind through that picture. The multi-author study found a startling pattern. Ectotherms, cold-blooded animals, display a huge diversity of aging rates, and many turtles, crocodilians, and salamanders show remarkably slow, sometimes near negligible, senescence. Protective traits, think the turtle shell, and low metabolic paces were repeatedly linked with vastly extended lifespans and reduced rates of age-related mortality. In short, cold blood can mean long life, not because of magic, but because physiology and ecology reshape the evolutionary trade-offs that normally favor fast aging. The mechanisms behind slow aging: protection, metabolism, and life strategy. The study highlights several recurring themes. Protective phenotypes. Hard shells, armored skin, or inaccessible habitats lower extrinsic mortality. If you're unlikely to be eaten, there's evolutionary room to invest in a long lifespan. Low metabolic rate. Ectotherms often run life at a cooler pace. Lower metabolic turnover appears linked to reduced molecular wear and slower accumulation of damage. Slow life history. Long development, infrequent reproduction, and extended parental care all co-evolve with low aging rates. When reproduction is slow, selection favors maintenance that keeps the body functional longer. These aren't metaphors, they're testable hypotheses. The reptile/amphibian dataset gives researchers a map. Compare species that share environments but differ in shell, or that differ in metabolic rate, and you can see how each trait nudges the aging curve. Longevity emerges from ecology plus physiology, not from a single immortality gene. From desert tortoises to sea giants, why the deep is a loneville. If ectothermy is a first hint, the ocean, especially cold, deep water, is an absolute laboratory for slowness. Low temperatures, stable conditions, and sparse food favor creatures that grow slowly, reproduce seldom, and invest in durability. That helps explain how some marine animals reach centuries of age, and none illustrates that more dramatically than the Greenland shark. Greenland sharks, the centuries-old monsters of the Arctic. These pale, slow swimmers of the North Atlantic live at the chill edge of the world. They grow less than 1 centimeter per year and can reach lengths exceeding 6 meters. But the remarkable fact is their age. Using radiocarbon dating of eye lens nuclei, a method that reads the chemical signature left by global nuclear testing and maternal diet in the protein core of the eye, researchers estimated Greenland shark ages spanning centuries. In a sample of females, ages ranged up into the multiple hundreds, One female's estimate approached 500 years, making Greenland sharks the longest-lived vertebrates known. Their secret appears to be a metabolic economy. In freezing Arctic waters, biochemical reactions slow. Growth is glacial, damage accrues slowly, and reproduction is paced across lifetimes measured in centuries. These sharks are not immortal, but they are near timeless by human standards. Ancient bristles, black corals, and the tree-like architects of the deep turn deeper and older still to the corals that wear black skeletons. Black corals, Antipatharia, are not your bright reef corals. They are slow, tree-like animals that have radiated through deep water and, according to paleontological and molecular clocks, belong to a lineage stretching back hundreds of millions of years. Fossil finds push Antipatharian relatives into deep time, showing a capacity not only for extreme longevity in individuals, but for evolutionary persistence across mass extinctions. Their strategy is patience, slow skeletal accretion, filter feeding on sparse plankton, and occupying niches where competition and temperature shifts are muted. The result is longevity on two scales: individual durability and lineage robustness. Recent genomic work is beginning to reveal the molecular toolkit these corals use to resist damage and repair tissues over geological time. Silica cathedrals, glass sponges that survived ice ages. If black corals are ancient architects, glass sponges are cathedrals in slow motion. Species such as Anoxicalix jubini, the Antarctic giant volcano sponge, build silica skeletons that persist through millennia. Age estimates for some Antarctic glass sponges range into the thousands, even tens of thousands, of years, making them contenders for the oldest individual animals on Earth. Their secret is simple ecology. Frigid, stable waters and extremely slow growth create organisms that trade speed for endurance. They are ecological time capsules, survivors of multiple glacial cycles, repositories of ancient microhabitats, and hosts for specialized symbionts and crustaceans that rely on their spongy scaffolds. When you walk among them in imagination, you are stepping through a living fossil forest. The jellyfish that restarts life. In the quiet waters of the Mediterranean and the Pacific drifts a creature no larger than a fingernail, Turritopsis dohrnii, often called the immortal jellyfish. At first glance, it looks unremarkable. A tiny bell, fragile tentacles, a body made mostly of water. But under stress, starvation, injury, sudden environmental change, this jellyfish performs something no other known animal can do. It reverts. Instead of dying, it collapses into a blob of living tissue, sinks to the seafloor, and reorganizes itself into a polyp. The juvenile stage of a jellyfish's life cycle. From that polyp, a new medusa buds, the same individual, a new beginning. This is not reproduction. This is a biological reset. The process is called transdifferentiation. Adult cells abandon their identities and become something else. Muscle cells become nerve cells. Skin becomes gut. Structure becomes a possibility. Discovered in the late 19th century but only fully understood in the 1980s through laboratory observation, Turritopsis dohrnii does not age in any conventional sense. It can repeat this cycle indefinitely, at least in theory. It is not invincible. It can be eaten. It can be destroyed. But it does not have to die of age. In nature, it likely restarts multiple times before predation ends the cycle. In the lab, under protection, the loop can continue again and again. It is the closest thing biology has ever produced to functional immortality. These animals did not defeat death. They simply negotiated better terms. They show us that time is not a universal tyrant. It is a pressure evolution can reshape. Some species live fast and burn out. Others barely move and endure. If you want to explore the creatures that rewrite biology, the science that questions what life must be, and the hidden systems that shape our future, subscribe to Discovering the Unknown. Because the unknown isn't waiting at the edge of the universe, it's already alive, and it refuses to die.